Good evening, everyone. Once again, thank you for joining us. It is the last session for this course, and the reception and as well as the feedback has been very positive. We had an opportunity to hear from pioneers their experience. Maybe very nice to talk about their insight and mistakes, which we should not do it in future. With that, I will open the session. First session will be taken care by Dr. Hemant Malhotra sir. Sir is currently working in Mahatma Gandhi Medical College, and sir will be talking about calculating compensation for participants of clinical trial. Over to you, sir.
Thank you very much, Vinayak. If I may be permitted to share my screen, here is my screen. And I'm going to be talking today on issues related to compensation in clinical trials, the calculation part and the compliance part. And this is my medical college hospital. My most sincere thanks to Dr. Vinayak and Dr. Parikh for this opportunity. So I'd like to start with a disclaimer that the views which I'll be expressing are essentially mine and do not reflect the views of my institutional review board or the DCGI or the regulator. So that kind of disclaimer has to be kept in mind. So ladies and gentlemen, I came across this publication, very decent publication, just less than a week old, in which in the JAMA Oncology, these gentlemen have analyzed randomized clinical trials and compared these published randomized clinical trials between low and middle income countries and high income countries. And we have some important investigators from India. Dr. Pramesh is here, so we can assume good fair participation from India. And this is some of the findings they have out of a total of 121 randomized trials in breast cancer. Only 6 trials were from LMIC and India. And similarly, if you see head and neck cancer, which is the #1 problem which we have in our country, out of a total of 34 trials, only 10 trials were from India. Another important factor which came out from this publication and this analysis was that impact factor of the trials in low and middle income countries was 0 to 6, while for high income countries, it was, on the other end of the spectrum, 26 to 80. And these were the crux of the findings from this study, that out of almost 700 trials, these publishers, the authors found that they were conducted predominantly in high-income countries, and this does not match the cancer population vis-a-vis global burden of disease. And even though randomized trials from LMICs are likely to result in new treatments proving or disproving benefit, these trials are not published in high impact factor journals. And this is the conclusion and the relevance which they quoted, that this study suggests that there is a funding and publication bias against randomized trials led by low and middle income countries. And policymakers, research funders, and journals need to address this. And this includes capacity building. And I'd like to really, there was a very beautiful editorial also accompanying this publication. And they also kind of highlighted on the need for resource development. And I'd like to, thank Dr. Vinayak and Dr. Parikh for this great initiative, which is, I think, very important for resource building for doing more and more good quality randomized clinical trials in our country. So with this background, I'll launch into my topic. A little bit of background, we had the golden period, so to say, of clinical trials in India in the 1990s and the early 2000s, in which, and the advantage of the large number of trials coming to India, we knew that there were a large and diverse treatment naive patient population. We had trained human resources. We had good clinical practice, compliant investigators, and relatively low cost of conducting trials in our country. But post 2010, this bubble actually burst. And this was predominantly because of isolated cases of incorrectly conducted trials, not conforming to ethical principles, and also quite largely because of unbiased and one-sided media reporting. And this led to massive debates in the parliament, in the public, several PILs by NGOs in the Supreme Court. And as a result, like this golden period of 500 plus trials in 2010 fell to about 100 trials just in a period of 3 years. And consequently, this also led to the drop in the number of new drugs approved for marketing in India, 200 plus in 2010 and only 35 in 2013. There was this public and multiple public interest litigations in the Supreme Court. The prime concern raised was that the informed consent process was not properly followed, and compensation to trial participants were not appropriate and as per what they should be. The government of India then filed an affidavit in the Supreme Court in which they listed that between 2005 and 12, 2,868 trial participants died, and compensation was paid to a very small minority, and the amount of compensation ranged from 55,000 to 42 lakhs. And this amount of compensation was not based on any objectively defined guidelines, but was very subjective and in the assessment of best judgment of ethics committees or sponsors or investigators. So from the Supreme Court, there was a big hue and cry, and this led to the government mandating a three-tier system of clinical trial approvals and several modifications in the DCGI Act. And this led to 3 landmark amendments regarding to compensation in clinical trials, conduct of clinical trials, and registration of ethics committees. And all of you are aware of the dates of these. Again, the Supreme Court mandated that justifiable compensation to participants who suffered trial-related injury or death is to be viewed as one of the important areas of clinical trials. And again, a notification was issued in April in 2014. And these were the main points of this notification. And some of them are reasonable, and some of them, particularly these 2 points, C and D, did appear to be a little unreasonable to some of the investigators, like failure of investigational product to provide independent therapeutic effect, and use of placebo and placebo-controlled trials. So again, this led to a lot of hue and cry. The pendulum, which had swung completely to one side, some corrective action happened, and some, this was a kind of an attempt was made to fix this, because, how can you expect an investigational product to provide benefit in all cases? That is the reason why you test that investigational product. So some modifications were made. And then, these principles were adopted in the clinical trial compensation document. The philosophy of compensation in our country as of today is what is known as no-fault compensation, and this has been adopted in our country. This actually means that every injured participant gets compensated, even if the injury was anticipated, expected, and even if the patient was fully explained that this could happen in the informed consent process. The trial participants or their relatives did not need to prove that it was anybody's fault, did not need to go to the court of law to seek compensation. Now when you compare this with what happens in the rest of the world, in some developed countries of Europe, France, Spain, Belgium, the compensation with clinical trial is also provided on a no-fault basis. However, most of these countries do not have a compensation formula. And the method of determining the quantum of compensation varies widely and varies from case to case. In the US, again, very large country, so to say, developed country, they follow the torts law, which means that if compensation needs to be paid, then it has to be proven in a court of law that the trial-related injury or death occurred due to fault of either the researcher or the research process or the protocol. So even free management of trial-related injuries is not mandated in the US under federal laws. Now coming back to India, to ensure objectivity and applicability in diverse circumstances, the best fit formula incorporates the important factors like age, baseline risk for determination of compensation. And the final formula, which again, I think most of you are aware, is 8 lakhs multiplied by age factor, multiplied by risk factor with a division factor. 8 lakhs being the base amount, age factor varying from 22.2 to 8.54 if the patient is very young, to 99.37 if the patient is on the older side. And the risk factor ranging from 0.5 to 4, again, determined on the basis of the chances of survival at the time of enrollment. And, I was part of this committee which decided this factor, and we had a lot of to and fro's and ups and downs. And, like I can take credit for this division factor over here, because like some of us thought that if this was not there, if the amount of compensation was too high, that could be that could serve as an inducement for participating in the clinical trial, which would be against the principles of good clinical practice. So according to this formula, minimum compensation of 2 lakhs and a maximum compensation of 73 lakhs can be awarded to participants in the clinical trial. So this is the basis on which the compensation in our country is based. And I'll just go through a few cases. I was also on the phase 5 SAE committee of the DCGI. Some people used to call it the death committee before we asked them to change it to a grade 5 SAE committee. So let's talk about a few peculiar cases which we faced and saw and decided on in this committee. So let's talk about case 1. This was the study, a randomized open-labeled phase 3 study of erythropoietin versus standard supportive care in patients who have anemia and who have metastatic breast cancer receiving standard chemotherapy. So we had this 39-year-old patient, date of consent was March 2010. The patient had an SAE several months after initiation of treatment. Patient was on the standard supportive care arm without the investigational product. The patient had seizures associated with altered sensorium and death. Date of death was just 2 days after the patient was hospitalized. And, EC submissions and, everything was then done. And compensation in this case was not given. So this is the details of the patient. Patient was on chemo with paclitaxel gemcitabine, received the last chemo about 10 days before hospitalization. Patient came with seizures and altered sensorium. Clinical diagnosis at the time of hospitalization was a CA breast with CNS metastasis. Everything else, all of the causes of seizures were ruled out by biochemistry. Other than a little raised liver enzymes, everything was normal. MRI scan was planned but could not be done because of the patient's poor condition. She could not be shifted to the MRI room. Appropriate treatment was instituted, both from the point of view of infection and from the point of view of increased cerebral edema. Anti-epileptics were given to the patient. In just 2 days' time, the patient's condition did not improve and she died. So if the opinion of the PI and also the ethics committee, because the admission diagnosis and the possible clinical diagnosis for brain metastasis compensation was not given. This case did come up to our committee from the DCGI, and we concurred with the PI and with the ethics committee that this is probably disease progression and compensation need not be given. So sometimes situations are clear, the chronology of events is clear, and sometimes it is not clear. Let's talk about this particular patient because the first possibility was that the patient had CNS metastasis, which was because of disease progression. Compensation was not given. So let's talk about another patient. This is related to a trial which is a randomized double-blind placebo-controlled study to again evaluate the efficacy of darbepoetin in anemic subjects, this time patients with non-small cell lung cancer receiving chemotherapy. So now we had a 57-year-old male patient, retired farmer, stage 4 lung cancer. He was consented, recruited, started on chemotherapy. And because this was a double-blind study, we did not know, investigators did not know whether the patient was on the darbepoetin or was on placebo. So after receiving his third cycle of chemotherapy, 2 weeks after this, the patient died at his home. All the interrogation was done on the telephone, talking to the patient's son. And the patient's son did disclose that the patient had dyspnea a few days before death, and no medical attention was sought, and the patient died at home. Now the causality assessment, the PI thought that it was not related, sponsor thought it was not related, ethics committee thought it was possibly related. When it came up to the DCGI committee, we thought that it could be related. The patient had a definite history of sudden increased shortness of breath. One of the important causes of all the erythropoietin stimulating agents is an increased tendency to pulmonary thromboembolism, and it possibly could be related to the investigational product. So what we did was, because this was a double-blind placebo-controlled study, we asked the sponsor to break the code and provide to us and tell us whether the patient was on the IP or was on placebo. The sponsor came back to us and told us that they would prefer not to break the code on scientific grounds. And after several weeks of back and forth between the institutional review board and the sponsor, this matter came up to the DCGI, and then we decided that we will presume that the patient was on the investigational product, and because the investigational product is well known to produce thromboembolism, we suggested that the patient be given compensation. So the compensation of 2 lakh rupees was minimum compensation was given to this patient, because this patient was elderly and he was not actively employed at the time of inclusion in the trial. So this kind of concluded again, and this again brings up several issues about these double-blind placebo-controlled trials. If this patient had a SAE and had been admitted to any one of the hospitals, it would have been mandated on the sponsor to break the code and tell us, and tell the investigator, tell the principal investigator whether the patient was on the placebo or was on the investigational product, because that would impact on the patient's management when he had the SAE. But here that issue is less important because the patient died at home. But then again, this is an important issue which deserves attention, which deserves thought, that for placebo-controlled trials, the causality assessment sometimes can be tricky and sometimes can be difficult. Last case before I conclude. Suppose this patient, the same patient, exactly same scenario, same study, this patient had been a middle-aged or a young female, a homemaker, right? Everything else being the same. This is hypothetical. Now, in this situation, if the case came to you, how much would the compensation be? Would it still be 2 lakhs? Or because the patient is 42 and female, would it be different? So I just brought this up because very recently in the newspapers, I don't know the legal situation over here, but in the newspapers very recently, again, just last month, this issue was highlighted, which I found was very interesting. Conception that housewives do not add to economic value of the household must be overcome. And this case was discussed in the Supreme Court. This was basically a lady who died in an accident. The one court awarded 44 lakhs of compensation to this lady, and another court reduced that to 22 lakhs, saying that the lady was a housewife and was not earning money, was not in service, was not in a job, didn't have a salary, so she should not be given that much compensation. And the Supreme Court, after all these discussions, came to the conclusion that no, all these factors came up, and the Supreme Court then said that the homemaker's notional income must be determined by courts, keeping in mind the number of women engaged in this activity, the value of their labor, services, and sacrifices. And this activity should be calculated in terms of direct economic value to the family, and compensation should be calculated on that. Supreme Court then in this particular case changed the compensation again to like 44 lakhs. So this is going to come up in clinical trial compensations again when you have a homemaker having an SAE and a serious SAE. How do you calculate the income of the patient? Does the contribution which the homemaker make to making the home, does it have a direct economic value? Can that be translated into an economic number? So this again is food of thought for our delegates. So if I were to give you my stay-at-home messages, we all know that clinical trials are essential for progress in medicine, and the safety of clinical trial subjects is of utmost importance, and even more important for our subjects, who in the low and middle income countries, they are relatively less informed as compared to their counterparts in the developed world, and it becomes the responsibility of every principal investigator to even be more careful than just the documentation which is required. An optimal compensation for clinical trial-related injury or death is difficult to determine. It has to be a delicate balance between inherent risk in the scientific pursuit for patients' benefit and safety of the trial participant by virtue of their participation in the clinical trial. And regulatory guidelines are part of a dynamic process and will continue evolving as per the changing requirement and demands of the system. As you know that initially those two points which were included in the DCGI document, they had to be partially modified. So with that, I'd like to close my presentation, and if there are any discussions, I would be more than happy to contribute.
Thank you, sir. That was a very insightful presentation, and I would open the floor for any questions. I don't see any questions except for Dr. TP Sahoo's question that any data for approved for in 17 to 20. I don't think anyone will have the answer, but I'll. Any data for it was very interesting slide, sir. When we saw that how the numbers came down. Fine. I will plan next session, sir. We'll keep questions in the end. I would call Dr. Yashesh Mehta from Mumbai. So it will be perspective from CRO and sponsors, how PI can do a better job for the trials which they have been assigned. Dr. Yashesh, can you hear me?
Yes, I can. Yeah, thank you.
And in session, sir.
Okay. Shall I put up my slides and initiate?
Yeah, you
can. No, sir, please go ahead, sir.
All right. Thank you. Give me a sec. Can you see these?
We can see, sir.
Okay, great. So good evening, everybody, and thank you, Kavina Creations, for inviting me to share some of these experiences on how PIs can do a better job. Here is the agenda. I'll be brief and quick and then open it up if there are any question and answers. So one slide essentially on key expectations, which is a little more theoretical than expectations, and then a couple of case studies. I thought it was easier to bring in case studies coming right from the fraternity and use them for these sharing instances. Quick disclaimer, which is important to know, these contents are my own understanding and experiences over the years. These are not related to the company that I belong to. Quick understanding on expectations, and some of the challenges. The challenges that investigators face, which is on the right-hand side. I'll go there first. These are standard. You have very high patient population that you cater to. Patient treatment priorities are your topmost requirement there. Besides that, there are site or hospital-related priorities. Meetings, pricing, rest of the nuances which come with the whole environment there. That's the second challenge, and then the other challenge is having trained manpower. So these are the three things which I could think of. And on the other side, despite all of these, the expectations that sponsors, CROs have from investigators are, we go to the busiest of the practitioners and expect time from them and approachability for conduct of studies and answerability on those. Ethics committee communications are some of the, and when I say expectations, these are also some of the challenges which we see when the studies get initiated. Ethics committee communications, contracts and legal handling. Enrollment is the expectation that the study starts on time. Studies enrollment happens on time as per the plan. Overall compliance, so that the data coming out is something which is evaluable and useful. Overall meeting timelines of reporting data, CRF entries, SA reporting, any other responses to communications, all of that. And then adapting to various dynamics that come up. Generally the studies are pretty long duration, and there is a need to adapt to, and like in the last year we had the pandemic, and there was a need to adapt to those kind of situations. So these are some of the expectations which make us decide on better investigators. By the way, some of the attendees here have been investigators to me, and I've learned from them. So happy to come back and share something. So some of the case studies that I'm going to share, that will be the exact thing which we have got from the fraternity. So the case study, first case study, the indication was gynecology. We had 6 sites, and then bringing in this lockdown time, there was one site which, so this is the positive side of the first study conduct and the PI's efforts. The patients otherwise would have been discontinued, but the investigator went out of the way, looked out for local labs which could help the patients go to get some tests done over there, and ensure visits were continued and the safety was evaluated, data was brought in. They also introduced some other facilities. I'm being quick here, but just understand that the out of the way that the site went. Taxi ambulances to help these patients to commute, come back to the site for essential visits and get the visits done. And of course, the sanitization precaution, all of that. The benefits which came out of this was we minimized protocol deviation from this site. And whereas we had lost around 17 patients from other centers which participated, this site lost, saved 6 patients which were there on the study and added 2 more. So that's the benefit that we got from this kind of a positive approach, a proactive approach that the investigator had. And the outcome is that the next 2 studies are going back to this center. So that's a very strong example of what the expectation is and how the investigators can do better. I'm moving on to the next. I'll go through these very quickly and then see if we can take questions. The second is also again a similar situation where during the pandemic, one of the sites which was a high enroller. I've got 2 columns here: site A, site B. Highest enrolling site, extremely hard working on that area. The pandemic called out for a travel policy where restrictions were brought in for monitors to travel. The investigator went on to help us out with performing remote data, source data verification. So they scanned some of these source documents, and there was a secure site which was from the site. and where we could do the remote source data verification. They also helped us to get these permissions from the ethics committees and lab reports which they had, so they could put them down into Excel sheets. So that helped us to do a continued amount of source data verification, and at the end of this lockdown, we may still have a possibility to do a on the site. On the other side is a site which, though not the highest enroller, could not really cope up to the requirement. There were constraints that were brought up on how CRC time would be utilized. There was a fee, but the site could not really do this part. So we could not monitor, this center for quite some time. So that's the other side of a site. So obviously, the example is that we would prefer to go to a site which is going to be more available. So here we went out to reward the CRC, and of course we are looking at the next study to be brought in here. Oh, sorry. The third case study, it's a combination, a couple of examples. I've just crunched them in. I'm going a little fast because there are 15 minutes and I wanted to cover as much as I could. There's a combination of events which we have brought in here. One of the situations where the clinical trial agreement, when you have to sign that, it goes to the site, and the utilization is of a senior CRC level person who reviews these. And our experience in this case, unfortunately, was that the reviews were a little outdated. It was difficult for getting anything from the legal units there, and that caused the delay in startup of the site, and the other sites went on to enroll. On the other side, another ethics committee where they brought up examples or suggestions to modify the informed consent form to recommend certain medical procedures, some of the other recommendations to just highlight certain sentences, bold caps. These are not ethical considerations. These were all procedures which were brought in. So here is a situation where handling of the ethics committee and convincing them also came in. The outcome is that we had to put this site on hold and go ahead with the other centers. These are examples which I thought were fresh in mind and we could bring up. Then the fourth is also again something which is a combination of things which we can look at on how investigators can be doing a better role. And if you see on the right hand side, what is preferred and what is missed. I think if we look at the couple of requirements or understandings from the sponsor CRO side, having a good next in line to the investigator so that the timelines can be met, solutions and understanding of the expectations is the first requirement, on which many a times can get missed out. Have some of the examples is what I just mentioned earlier in the earlier slide. Having a strong next in line so that you can have the medical part delegated out to co-investigators, but value adds coming in from the study coordinators in terms of prompt and thorough documentation, which becomes the base for the study to be evaluated during audits and inspections. Timely reporting of data, which could be into ECRFs, it could be into project management tools, it could be into SA reporting, and then of course the client communication part. So having a good hold on that is important. So in some places, what we also feel the investigator could have done better was a stronger oversight. So that, for instance, having a better hold to get the legal thing sorted out, or having a better communication line with the monitors, regular meetings with the monitors or the team leads is something which will be very useful when you are doing the investigator's task. So these are some of the expectations and what is preferred from investigators to do a better job is what I could bring out. Of course, professional dialogue so that you can conduct the study well. That is another thing which is important. I think that's the 4 case studies. I don't have too many examples or things which I should bring up on slide, but I'm open to questions if there are any.
Thank you, sir. It was very enriching that we saw case-to-case basis because more of a understanding. So I just want to understand with the COVID, with the clinical trial,
Yes.
what are the challenges as a CRO or as a sponsor you found in those times? Just before we go for the
Yeah. I think the 2 or 3 cases I just presented. The first one was that the personal touch and the travel to sites where we could coordinate and get work continuing is the thing which we missed out. Some of the sites had patients who had. I mean, the natural phenomena was that the patients were not reaching the sites or the sites were shut down. So enrollment had stopped and the patient visits were not happening. Both the things were something which we faced as challenges. So in these situations, one of the centers which I mentioned, they went around to help the patients, at least the ones who had already come onto the study, by helping them with these taxi ambulances, helping them to get a sonography done very near to their homes, and that helped the study to continue. These patients were not discontinued because of the follow-ups which they had to do, and that's where they saved discontinuation of 6 patients, whereas other centers we lost patients. So that's been a big challenge. Otherwise, gathering data, because many a times the coordinators, study coordinators who are operating at the site, they were either not available to go to the site, or there were confidentiality concerns which came up on how does the data come to us, because they have to enter it from the patient source file into the electronic CRFs. So, how do you do that? And if we have to verify during monitoring, how do I do a source data verification, source versus ECRF? So that's the second case where we gave an app to the center, and the site had. So what happens is that site has the rights to give access only to the monitor. It's not like we have access to the app. The site has the access, and they give the authorized personnel from the CRO or from the sponsor access to the source for a day. So if the monitor comes in, looks at the source, looks at the ECRF, and the verification could be done. These are some of the things which were challenges, how the solutions came about. So those sites which helped us with this, we went back to those sites and thanked them, of course.
So in this pandemic, we learned a bit of doing things differently. Is think anything will be continued in the future, like remote site visits and other things?
Yeah, it's a very important question. I think everybody has almost realized that the world is going to change significantly. A lot of these visits will, anyway, clinical trials were moving towards the risk-based monitoring. Good chances of having a certain percentage of remote monitoring which will come up in the near future.
Thank you.
Personal touch is required, but there'll be a decent amount of remote monitoring.
We should stay back. We'll ask more questions in the question answer. So we have 45 minutes. So I think there's no questions in the chat box. So I would go for next session. Thank you, Dr. Yashesh.
Thank you.
Dr. TP Sahoo, sir. Yours is the next session. And you'll be talking about common mistake and deficiency, which need to be avoided as a PI or as a site thing. Sir, I'm handing session over to you.
Okay. So my slides are visible?
Yes, sir.
Yeah, thank you. So this will again be a very practical thing, and this is what I have learned about how to go around things which is normally written, but some things are not written. So I'll be very, very practical. It's my own experiences. And I think these are the common mistakes which we do. And the topic given to me is the mistakes and deficiencies to be avoided. And I think the topic was for a principal investigator here. So the point is, for starters, the PI is responsible for everything on the site. So once you're responsible for everything, the PI has to have knowledge about everything. He may not physically do everything, but as the previous speaker really spoke about, is the term called oversight. And the person in the charge has to know what is happening to everything. Then only the CRO or the sponsors feel confident about the site and also coming back to the site. And most important, as we discussed yesterday, was to choose an adequately trained and sincere trial team. That's something very, very important. What many sites do sometimes, which I have seen during the days where I was very much active in trials, was they used to do most of the staffs were either hospital staffs doing a part-time job in trials. You can have that, but what happens is the mistakes will grow because there is a hospital responsibility. You are also part of the hospital, so you have to have dedicated patient personnels who only do clinical trials, who are part of your team, and this will be the study coordinators, as rightly spoken by the previous speaker again. They are the skeleton on which you really build your team. A good clinical study coordinator really makes the job much easy for the principal investigator. So let's go one by one where I thought I could come in and avoid the mistakes. And because I've gone through all the 3 audits, the FDA, the EMEA, and the DCGI audits, I can tell you what they really pick up and where the mistakes really happened. Luckily for me, I did not get any notice for any one of the 3. There were some minor observations which I feel could have become major if this was consistent across the patients. So what you look at is once a study comes in, once you're okay with the study, you really, this delegation log is so very important. Many times it is filled up just for the filling up. But what happens when the FDA comes in or the EMA comes in, even the DCA comes in? I've seen more with the European and the FDAs. They look at this delegation log very carefully. And anything that is given here, the person is not delegated, has done that thing, even for something as small as a vital recording, then that is pointed out. And if it is a pattern, they put it as a very major observation. So what I mean to say is be very clear about what you delegate to what person is delegated to what. Make sure the person is qualified to look into the delegation, the responsible delegated, and make sure what I have learned over the period is give little more extra responsibilities. There could be overlap of responsibility that makes your life easy, because then the problem with that this person was not there for delegation is not there at all. Next is the period. There is something called as involved to. Normally it is the end date where the site is closed, but sometimes if you close something and the person is just closed for something and then the patient comes back, there is something the person has worked after the particular date of involved to, it becomes a major violation because the person was no more a part of the trial. So that is something that happens when you backdate things and when you ask the person, maybe radiologist, to write something, and unluckily you have forgotten that this radiologist was not part of the team at this point of time. So that becomes a thing which you have to be very careful. This is a small thing, but this can lead to major problems when you are in the scrutiny of the regulators. Now come to the second one, inclusion and exclusion criteria. Because the basis of how we take the clinical trial and the patient into a trial is the inclusion exclusion criteria. What I have learned over the period of last 8-10 years and 40 odd trials, the trick is give adequate time. There are some 15-20 inclusion and exclusion criteria. Most of us remember some of it, not the accuracy of it. What I used to do after that, once you do the trials, you memorize a lot of things. Then after you're done that, when patient looks interested in a trial, you've spoken the initial part. What I used to do is give to my study coordinator to look into every point, tick every point. So what you need to do is give adequate time. And not every patient is a trial patient. This is where I burnt my fingers when I started trials. Every patient, you are so enthusiastic, you want to be in the race, you want to be the highest recruiter. So every patient that comes to OPD, you want to recruit. A lung cancer case, you want to recruit every lung cancer so that you are the highest recruiter and you get that applause from the CRO or maybe the sponsors. But I think over the period of time you learn that please, not every patient is a trial patient and you are not in a race. It is important to choose the right patient, right inclusion exclusion criteria, and keep the patient in the study as long as possible. So that's very important. And don't manipulate data. All of us will tell you, but I'll tell you what I felt is the most manipulated data is the performance status. It is so typical that what you write is how it is perceived. There's nothing to cross-check it. And all of us know in oncology, a PS2 can be made PS1. But you then get into the trouble when a PS3 sometimes is taken into the trials as PS1, and then the patient dies within a short time because the patient of these PS2, 3 normally do not survive for a long time. So this is probably the most falsified data, and this is my take on what I've seen from my colleagues and what I have learned. So in my later trials, I was very particular of the performance status. It has to be a 1, 0, or 2 if the trial wants to include them. Now, this is something which all of us spoken, the informed consent. All of us know what to do, but I'll come to the practicals of it. All of us prepared a checklist. Still, this is the most important thing. If in a, let's say the EMEA, I went for 3 days, they took at least half a day to look into my 20 consent forms. There was 20 patients into that multicentric study of a multinational company. They took at least 4 hours for 20 consent forms. That's how rigorously they look into the consent forms. And that is where the trick is. Because one thing the consent is wrong, you are really in a soup. You don't know what to do because it has been done. The trial is over. They come after a long time. What is the practical tips what you can tell my juniors who are here? Check the version. This is one of the most important thing which you should do. And check the version means there are multiple versions which would have come, which would have approved by the ethics committee. There are very minor changes. Sometimes you take a signature on a previous version which you should not be taking, because after ethics approves a new version, that's what is approved and you should be taking it on that. So destroy all previous ones. That's what I learned. Except a single copy for record. You keep that. It is out of bounds, all of you, because anyone would not have access to that, and you have to keep anyway one copy blank. But use the new versions, and that's how it should be. Make a marker for that. That's one of the very common mistake people do. Second, what people do is, and Dr. Malhotra briefly touched about it. I don't know how many people went into that, and I was lucky to get a very good things in this EMEA and the FDA for this. The document process should be very, very important of a consent form. You just do not write patient filled inclusion criteria and patient is fit for trial, agreed for trial, and do it. It's video recording can be there. Sometimes some trials only ask you to just consent it, and only the video recording of the signing part is only asked for. But most important is 2-3 things which I've learned is always document that you have given adequate time to the patient to think about before going to trial. Let's say the timing is important. You have to do ideally is the patient should not be coerced. All of us know. How do you do that? Important is you give a blank consent form, you speak to the patient, and the patient comes back after a particular period of time, maybe few hours, and then you ask what are the questions. That all has to be documented in the consent process. And important is the line which I've learned also, that all the possible treatments outside trials were discussed. That's something very, very important to write. So consent process for me in my later part of my trial used to be 1.5 page, maybe 2 pages sometimes. And that saved a lot of hassles for me. Timing. Never do any trial related event before the consent process is very, very important. Document a giving of a completed ICF to the patient. Once you've signed, everything is done. The photocopy is done. What I used to do, what people normally would be doing, give a photocopy and make the patient sign on it, receive a photocopy of the completed ICF. This saves a lot of trouble from the media when the things really get bad. Anyway, this has to be documented. It's voluntary, and give contact details of ethics committee and the investor team. It's there in the consent form. Sometimes it is missed. Sometimes the quickness of it, we are more into the signatures. This details has to be filled up. These are all what we are looking at. The version is in the lower part of it. The investigator name and a contact is there. Ethics committee contacts also comes in the trials. So purpose, all of us know. Alternative treatment documentation is very important. As a PI, you have to document it that trial was not the only option for the patient. You give the patient the other options. Compensation, we've spoken about it. Injuries already is covered. Voluntary participation, the word should be ideally coming in. So records. Now shift gears to records. Once you've done that, you've done the things. Records. Again, all of us know this. I'll come to the practicality. What does the law says? The investigator institute should maintain adequate, accurate source documents, trial records that include all pertinent observation of each of the site trial patients. Source data should be attributable, means you can attribute to anything to one thing and document it. Should be legible, should be contemporaneous, original, accurate, complete. Everything is there. It is legally so much strongly worded. Change to source data should be traceable. Do not obscure the original entry. All of us know that. So what I have learned. Document all the trial related work, because this is the most neglected part in a clinical trial from the site scenario. We do the lot of things. If there is a busy site, lot of patients on a day, you have 8, 10, 20, 30 patients, you have a limited maybe study coordinators, lot of the work is done, but the documentation happens later. Sometimes it keeps on pending, piling, that you start forgetting things. So ICF signature is the first of all of us know. Please document everything very, very clearly, and preferably the date and signature of every documentation is important, and no scribbling. You just have to put one line that is well known. Don't leave it for the last day. That is the only thing I have to say. If you don't leave it to the last day, you will not be falsifying data. You will remember everything. If you are very busy, at least take notes and complete it before you leave for the day. And at least, or you overlook that your site team has done that. ECRF, I got a little bit of minor deviation for this, that for the DCJ actually. The accurate time data is entered, that was tick for me. The timely manner entry they saw in few of the data, maybe 2 or 3, I had 1 or 2 days delay. They put it as an observation and told that you should not and you should avoid that. Otherwise, if it is consistent for the site, then we are going to put an observation for this. This is again one of the most neglected thing what I find is the temperature log. Many times you have many fridges, many refrigerators, many things to fill up. What happens is, this is used many times in many sites. I don't know now everyone must be doing it accurate, but normally what happens is sometimes it is sent black, and before a monitor visit comes, people start filling up. And I have learned over the time that monitors are very clever, and you as a PI also start to learn with that you cannot just do it the last day. I'll tell you why. Happens if you don't do it on a daily basis. And this is what it looks like. If you have done it, you can look at the slants. And I learned it over the period of a PI, having done quite a bit of trials, that if you do it, and if you do it in one day, the fatigueness starts coming in when you do the later part of it. You use the same pen usually, you use the fatigueness, and the slanting starts happening maybe on this way, one way or the others. So you have this slanting happening for the documents you fill up. You have the fatigueness of coming, the change of handwriting, and one single handwriting normally comes in. And that sometimes all of them look very, very suspiciously, because then you know they might not tell you, but then they make it as observation to look into your other things more carefully. So this is something which practically I've learned, that try to fill up, put a one study coordinator into the job is to come in daily, take the thing, as soon as they enter, take the temperature charts, because you always have thermometers which have been validated. That's make sure that validation is accurate. Yesterday also we heard FDA looking at the validations. Everyone looks into the validations, and this is a one-time job. Now this is important. Why I'm telling you, I had one it's way back in 8 years, 9 years back. I had 2 vials which was broken, and each of the vial was costing 2 to 3 lakh rupees of a drug at that point of time, not available in India. Breaking a vial is not a crime. Losing a vial is a crime. That's something all of us remember. If a vial is broken, please keep whatever is there. You are human. Your nurse might break it, but if it's broken, please keep that thing. Keep the broken things in a safety, so no one is injured. But let the monitor come, see that you have not misused the trial vial and it was really broken. I think then that is sorted out. That's something which I wanted to talk about, the accountability, the dispensing log and other things. I'll just skip the last part of it. As a PI, the adverse event and SAEs are very important. SAE, lot of things are spoken about. All of us know the timelines. I'm not going to spend the time on it, because SAE are so much important that very rarely people miss timelines. And that is something easy. But what I want to say from a clinician point of view is the adverse event. I have learned from one of my GCSF trials. I'll be very open that is the time when I learned that you have to be honest in trials. You have to give time for trials. It was GCSF trial from a multinational company. When I looked at my data for some 30 odd patients which I recruited, only 3 or 4 had myalgia or back pain. And when I looked at the data, when I just looked into, then I knew that something was wrong. I could not, or my team did not collect the real data, because if 30 odd patients have received GCSF continuously for 7-8 days, I would expect a majority of them having at least a back pain. That's what we normally see in our patients. So that's something which I tell SAE adverse event, although not as important as SAEs, you might make a drug look better when the end results come in. Document and inform is the ethics of any trial. Document whatever has happened, wrong, good, you will find a way to get out. The CROs really help you in coming out. The sponsors really help you out till you have a legal problem. And inform the authorities. That's something as the last part of what I would like to tell. These are the 5 key roles for the investigator. All of us know, and I would not spend time on it. For last part of it, hoping I've not missed anything. Apart from the last part, that as an investigator, you are responsible for the trial, running of the trial, and even storage of the documents. Yesterday we saw that FDA asked for the fire extinguishers. You have to make sure that these are fireproof cabinets and that trial documents will not be harmed. I can give you one prime example. I had left the site when the FDA audit happened. So I wrote back to the director or deputy director based in Mumbai, Mr. Scott, I think so. And I wrote, yeah, I have already left the site. So, but I would talk to the site and come back to you with the date, and I'll be available for you for the investigation. You cannot refuse them. So the next question he asked me, Dr. Sahoo, I want to know whether you have taken the documents with you or you have kept it in the site. Nicely worded, you won't feel anything wrong in that. So I had kept it there. I had sealed and signed it there. I told no, it is there in the site, well preserved with sealed and dated. So he wrote back, thank you for the reply. That's what I wanted to hear from the investigator. Ideally, you should not be taking these documents along with you. So that's something you learn over the period of time. And I think most of us have learned that. So conclude, to err is human. I think we have the CROs here. We have the other persons. We have Dr. Purvish, Dr. Hemant, who have done a lot of trials. To err is human. You will err. There's nothing wrong about it. But important is not to hide it. And if it is not a legal problem, I think everything is well saved and you can get out of it. Document, and that's possible. I think the only thing what we do is we normally, I've seen one basic difference. I can sound little more, I don't know how I'll sound here, but I've seen the basic difference what I found between the Westerners and us is we believe so much on our brain and the memory. Ye mere ko yaad to reh jayega mere ko. But they have something called as a tick mark thing. They write every small thing, and that's the basic thing. I still sometimes try to practice it, but many times I still believe on my brain more, and after some time I forget everything. So that's what is the bane of our being as PIs. Please document the same day before you forget, and it should be done in a timely manner. Don't lie and falsify data. I cannot just emphasize this as a PI. This is the most important thing. I told yesterday how a falsification of data led to the FDA blacklisting the site and the investigator. Every procedure is important. And the ICF, I can tell you, they spend a hell lot of time on ICF and the ethics committee approval. Once this part is done and you have not falsified data, I think you are through with every regulatory authority investigation. I don't think that is much you have to worry about. So with that, I really thank you for a patient listening, and I thought I could make it as practical as possible with what I have learned, where we do common mistakes, and as a PI we can learn from that. And one mistake can be others one that he or she will not repeat these mistakes. Vinayak, it's all yours. Thank you.
Thank you, Sahoo sir. It was very enlightening, and you have gone through a lot of audits and a lot of hard work. So it was very riveting to hear. With that, I will. Any questions? I don't see any question. Thank you. So we'll call our next speaker, Dr. Govind Babu sir. Sir is a mentor and guide. We rarely get opportunity to predict future, where we get to know how the science will go, how the things will go. Govind Babu sir will talk about how we are preparing for trials in 2025. Sir, over to you, sir.
Thank you, Vinayak, and thank you, Purvish, for asking me to talk on this. So what I've been hearing so far is all very practical stuff, what they have done and what they have learned from their clinical trials. What I'm going to show, I think, some of it is going to be fiction, but I think let's be prepared for it. So I'm going to talk to you about preparing for clinical trials in 2025. So it is said that if you don't plan for the future, then you're planning for failure. So we need to plan. So the first thing is we need to see what we are dealing with and only then. So the outline of my talk is going to be about the magnitude of cancer, what it is now and what it will be in 2025, because only when you know that magnitude can you plan and prepare for clinical trials. I will give you some tumor examples, how we have progressed, and I will talk about the evolving understanding of cancer and the technologies and how we need to adopt them and draw some conclusions. This is a very slide that you have seen very, very often, that heart disease is the commonest one and cancer is close by. But in 2025, I believe that cancer will be #1 cause of death. So this is just to look at the population distribution by age group from 2000 to 2050. And as you can see here, the variation that is happening is that the elderly population is going to be rapidly increasing. And this is this group that needs the most amount of support. And also, as we know, that this is the population that it's at the highest risk of cancer. And this is just showing you the same thing, that the increase in cancers in 2025 is going to be the maximum, whereas the others, cardiovascular disease, respiratory diseases, are going to be much less. This is talking about our own country. So the cancer scenario in India, today we have 1 million new cases detected every year, and 3 to 3.5 lakh die every year. And in 2030, we will have a population of 350 million, and the population that is more than 65 years will be over 20%. And we know that cancer is a disease of the aging population. So that is how much our burden of cancer in our country is going to increase. And this is actually showing you that the healthcare occupation, the projected supply and gaps in 2025, and this is in US, which we believe is one of the best societies. They themselves expect a lot of gap by 2025 in terms of the home health aides, nursing assistants, medical and clinical lab technologists, medical technicians, nurses, physicians, surgeons, and all others. So that is the gap that is going to be there. And this is an important issue that we need to address as well. And one of the important things we know as a problem that leads to cancer is inactivity. And this is showing you the global inactivity, how this is going to increase over the period of time. And you can see almost everybody will have highly inactive population. And this is just to tell you inactivity leads to obesity. And this is the next cigarette smoking, the obesity. So that is going to be a major issue contributing to cancer problem. And that is how we need to design our trials also better. And today we know that cancer is a genetic disorder and all the information is stored here in the DNA. And we are understanding this and moving towards personalized cancer therapy. And that's what our clinical trials are going to do as well. This is just a snapshot showing you how we have evolved in our therapies from 1996 to 2015 in these 5 major cancers. So we know that today each one of them has umpteen number of options that are there. And in 2025, I'm sure this is going to even increase further. So for a long time, I'm sure you'll recognize Mona Lisa. We thought lung cancer, breast cancer, colorectal, and many others were just one single picture like this. But today, if you zoom in, we know that each one of them is a collage of different diseases. And this is going to be an important aspect when we look at designing clinical trials, that we cannot put all these diseases together. We will be looking at subsets of these diseases, and we'll be designing clinical trials based on that. And we today have a classic example of non-small cell lung cancer, which is shown here, which is actually a poster boy today for these genetic alterations and the pathway-driven therapies. And this is what is going to be. This is actually just a lung cancer research landscape, which is looking at mechanism of action of various groups and the phases. And this is how our clinical trials are going to be designed, looking at the pathways and the drugs that are going to act on these pathways. And I think other thing is going to be looking at mutations. This could be one of those things, mutation, looking at tumor burden and the PD-L1 expression, which is going to be tumor agnostic. And this is one of the ways that we will be designing clinical trials in the future. We have always been looking at doing tissue biopsies to actually look at a diagnosis and enroll these patients on clinical trials. But I think liquid biopsy is making inroads into this, be it for diagnosis, subtyping the tumors, treatment decisions, and very important monitoring, because this is a very important aspect in our clinical trials. And this is already happening. This is an example I've taken to look at T790 cell-free DNA load to actually assess the therapy in terms of its monitoring and make adequate changes And I think this is one of the ways that our new clinical trials are going to be determined. I would give a similar analogy what we are doing today for chronic myeloid leukemia. We're looking at the BCR-ABL mutation burden and then trying to tailor therapy for them. I think this is going to happen to almost every tumor that we're going to treat in the future. So again, this is some work that we have done in looking at circulating tumor cells, and I must thank my collaborators for that. We have actually shown that, I'll just take two examples. This is to show the response of to gefitinib in non-small cell lung cancer. If you look at the orange bar, this is for what we classified and characterized as tight clusters. If you see earlier, these are small percentage of these bars. And once they developed resistance to gefitinib, this bar increased. And we showed that with a nice correlation with imaging. And once the patient was shifted to chemotherapy, these decreased again. So I think this is an emerging way that we will be looking at monitoring not just cell-free DNA, but the circulating tumor cell clusters as well. And this is another example to show you the same. And we have actually shown the correlation of these clusters, although small cohorts, but a good correlation with the clinical outcome, and we've also published this. So I believe that this is one of the ways that is going to be the future, how we will design clinical trials. And just last week, we had an international conference on liquid biopsy, and this was discussed as length as to how this is making inroads into designing new clinical trials. So the other example is breast cancer. I don't need to tell you how we have evolved in our understanding of this disease. And today we are looking at the genomic typing, and we are having extensive genomic analysis being done in breast cancer. So our eligibility criteria and the design of the trials is going to be based on genomic classification of these breast cancer tumor types. And that is how our new trials are going to be designed. So what could be the future directions? Like I said, the genome analysis is going to become an integral part of analyzing any tumor. And this is just to show you how the uptake of this analysis has increased over time and how the costs have dramatically decreased, making this extremely affordable for everybody. And I think this is going to be the way that we will be looking at inclusion exclusion criteria for our subjects in the future clinical trials. This is just to show you that over the last 20 years, therapy options have dramatically increased, leading to treatment complexities. And I've just given you some examples of some tumors and showing you how many options that we have. So in the future, this treatment decisions is going to be highly complex. And we already know today artificial intelligence and machine learning is rapidly coming into it. And I think this artificial intelligence will definitely come into the design and the integration of it into the running of clinical trials in the future. So one of these very interesting papers that was published was the tumor evolutionary principles and how intratumoral heterogeneity influences cancer treatment and outcomes. And this is looking at the tumor fitness and patient survival and the genetic instability and heterogeneity over time. This is just to show you how the tumors can evolve. This is a neutral evolution. This is a branched evolution, that is over time, different clones can develop. This is a linear evolution that several clones simultaneously develop during the progression of tumor. We could also have what is called the convergent evolution or parallel evolution. Now, we could have primary cancer with subclone interactions and metastatic cancer with subclone interaction. So this is a very extensive and nice way of looking at various evolutions of tumors, and all this can be done with the liquid biopsy looking at ctDNA. Now, how is this going to help us in our clinical trials is because looking at these changes that occur, we could look at adaptive therapy. We will design what are today called as adaptive clinical trials, that over a period of time, as we see the changes, we're going to adopt our trials. And this is how our understanding of the tumor evolution in terms with shown by the circulating tumor DNA is going to play a part in our clinical trial design of the future. Or it could be a sequential therapy. And that is, you look at what happens here, start a therapy, follow this tumor, how it evolves, and then change your therapy. And the classic example is what we have been doing in non-small cell lung cancer with the EGFR blockers. So I think these are the ways that we are going to rapidly evolve in the designing of our clinical trials. And this is a very nice, actually a market clinical trials survey that was published. And this is talking about all the diseases, including oncology. And this says that India is going to play a very important role in these clinical trials, especially in oncology. There's a very nice paper if you want to read in more detail. I would definitely recommend you to this. And this talks about the various phases of clinical trials and how it is going to change and the forecast for 2025. And this is from what is called the Future Watch, how the digital world is going to be evolving and how it is going to impact our clinical trials. We are going to look at alternate care locations. And as you know that the aging population is going to increase, we're going to have what are called smart homes and smart hospitals. And this is going to be because we today have enough gadgets that can give you all the data that you require, and the patients don't need to even visit your place, and all these technologies are going to help you to design your clinical trials better. And therein comes the smart hospitals. And we always thought the smart hospitals are digital, but that is not true because that is only a part of the smart hospitals. And this is how our smart hospitals are going to be. And based on this, again, we need to design our clinical trials better. And this is again showing you how we will be using devices, various softwares, various services in our hospitals. And accordingly, we need to take into account all these when we design our clinical trials. So we're going to use more and more robots. We may do away with many of our service staff. We may do away with even our clinical trial assistants, and you're going to have surgical robots, medical robots, and personal service robots. All these need to be kept in mind when we are designing clinical trials, because I'm sure with all this, the efficiency of clinical trials is going to improve, and all the mistakes that we have been making so far, which all of us have been discussing in the last 4 days, can be taken care of. So with the therapies that we have, where are we now? We are somewhere here. But where we want to be is somewhere here, that we see more and more patients improving and living longer. And for that, I think we need to design our clinical trials better. Molecular-based therapies are the things that are going to be there, and empirically determined therapies will be extinct like dinosaurs. And I would like to just show the last few slides. This is what our OPDs used to be when we started practicing medical oncology. And then it's become like this. But what I think in the future is going to be pathway OPDs. And that is where we need to be prepared for. So how are we going to prepare for 2025? As I've shown you, to estimate and anticipate the magnitude of cancer, augment manpower, technology, etc., design our clinical trials better. And one of the ways is to have adaptive trials. Can we do away with classical animal models of drug testing? Yes, possible. And we have done some work on ex vivo models, and this is coming up in a big way. We are actually now culturing the circulating tumor cells, and we're trying to see if we can use that for drug testing and drug development and adopt better technology, including the robots and the artificial intelligence. With that, I will stop and thank you. A lot of what I've told you, I think, sounds even to me as fiction, but I'm sure that it's going to rapidly come in, because when we started, I'm sure Purvish will remember, in 1990, we had about 5-6 drugs to treat. Today, we are getting 5-6 drugs every day to treat, and the inroads that the technology has made today into oncology is mind-boggling. Thank you very much.
Thank you, sir. It was gazing the future was very. I mean, where we will be placed in the future is a different question. But at least get treated by such drugs is a wonderful thing. And I think I remember an incident in our hospital when the COVID duties, there was little receptivity was not there among the colleagues. They were asking for robotic nurses to go inside other things. So I think we are almost there. So with that,
that's right.
So and I think the tumor cyto almost like it looks like a culture actually. That's the way forward I feel to test new drugs pathways. and we will have more of the drugs. Maybe we're going to culture each patient's tumor cells and get a product according to it. Sir is asking, Sudeep sir is asking, how are the prospects of home clinical trial? And in US, it's happening, especially during COVID times.
Yeah, in fact, I think to some extent we have also adopted it because some of our patients were not able to come. So we actually asked them to do some of the basic things at their place and take a picture and show it. Of course, the imaging and all, I really don't know because images has to be done in the hospital where the facility is recognized by the sponsor. So I really don't know how that is going to happen. But I think what I've shown and what I got from that future watch is really that everything can be done at home. So I think it is going to happen.
I have one question, sir. What future you see for regulations, ethical and other regulations, way forward?
I really, frankly, to tell you, I don't know.
Because there are a lot of issues with genetics.
Yeah.
We have to get regulation that people are not discriminated against genetic testing.
Yes,
The more
absolutely.
we go for method-driven thing, the more we will know that people are susceptible for disease.
Yes, yes. The classic example is BRCA today. Yeah, that's true. I think we are evolving on those aspects still.
Yeah, but it will be a very interesting phase to see a rapid expansion on all the fronts, and we don't know where we are heading. Sure, thank you, sir. I will ask a few speakers to stay back for the session. There is a panel which I'll be conducting, moderating the panel, and we have a lot of thoughts when we, as a PI, which trials we need to take, and sometimes we feel in the middle of the trial that why did I take, and those questions will always arise as a PI. So with that, I would ask few of our colleagues, few from the CRO background, few from the let it be from the radiation background, and from medical oncology. So shall I say share a screen?
Sir, only part of the screen is visible. Yeah, now.
It's visible?
Yes, sir, it's visible now.
That's right. Thank you. So the dramatic progression in clinical trials have made exciting outcomes, and we are giving a better and more effective drugs. But the question rises that all are fine. What is there for me? So in the minds of investigators is always whether to accept a declining clinical trial. You don't want to decline a trial because you want CRO to not get a perception that he is not susceptible, and you want to accept the trials which are reasonable to do at a time which is required. So esteemed panel, Dr. Aditi Parekh from Mumbai. Ma'am, can you hear me?
Yes, sir. Good evening, sir.
And Dr. Harshil, can you hear me? I think you're mute, Harsha. Someone needs.
So you'll have to unmute your mic, sir.
Dr. Harsha, can you hear me?
Yes, I can hear you. Thank you, Dr. Vinayak.
Thanks, welcome.
Yeah.
So Dr. Harshil has been a person who is from radiation background, but he has been doing a lot of clinical research, and he has been involved with India Gene and other things. So that I was really keen to him have to have board. Dr. Vineet Gupta. Dr. Vineet, can you hear me?
Yes, you are audible, sir.
Dr. Vijay, last to join.
Yes, I am audible. You are audible.
So we did an initial survey where we checked the PI's perspective. As we see, most of people are coming from a private, semi-private or trust background, less of in corporate and little less of in the things. So I would start with Vineet. Can you have it? So how was your experience as a PI as a first trial, as a principal investigator?
Yes, sir. So, sir, fortunately, I had experience as co-investigator in few trials before starting my first trial. So the trials which I was able to select, I had a good experience. But unfortunately, when I joined a new center, the previous consultant who had left the center, he had taken up a study as PI, and that study had to be inherited by me. So in this study, actually, I was not able to, service the study as well as I would have liked to, and the patient population was little bit difficult to get by. So I kept having a lot of queries again and again from the sponsor regarding why we are not able to, fulfill our promises. But I was not the person who made those promises. Regarding the other studies where I was PI, fortunately, I have had a very good experience, and I have not had a lot of problems, sir, in the other studies.
So, did any trials got you better learning than the trials which you've done before? Or first trial was
Uh,
fine. I would, Dr. Vijay, can you join us?
I'm here.
Same questions. Which was your first study and how did it happen?
My first study, my first 5 studies were actually investigated initiated, so they went pretty smooth. My first pharma study didn't recruit at all, and the reason was I learned it hard way that uh, multidisciplinary trials, which involve surgeons, radiation, and then coming to medical oncologists, are very difficult to handle, especially in government centers. And what the study was actually about giving a therapy post completion of adjuvant chemotherapy in lung cancer, and the patient had to wait for 1 year in Bombay. And up till now, I must have counseled at least 45, 47 patients, but not a single patient said yes to wait for 1 year after completion of surgery, adjuvant chemotherapy in Bombay. What I could do is that there were centers in other cities which were near to that patient's native. I actually sent the patient on for that study. In like if the patient was from Vellore to Ashish at that trial, so I'd send it to Ashish. If in Kolkata, if someone had, I'd send it to him. That's the way I did it. But I had it was pretty, and then after that I have been very careful for selecting trials. I actually do it this way that if I find 100 eligible patients, I divide it by 10 and then I divide it by 2. If that number is coming anywhere between what the sponsor is accepting, then I say that okay, that this will be feasible. Because in routine practice, you think that you may have multiple patients, but quite a few of the trials are quite stringent criteria, as Dr. Sahoo also pointed out. Performance status is not there. Sometimes the HB is just 2.2 below. Uh, uh, if the patient has a osteoarthritis, his PS may not be that good, but the sponsor is not ready to say that the PS is poor because of osteoarthritis and allow the patient to be taken in the study. So when you go to the stringent criteria, actually in pharma studies, you land up with a few patients. After that, the few studies which I took for pharma studies, which were mainly phase 4 and phase 3, which were common. So they recruited well. In fact, they recruited better than what was the sponsor's expectations. But my first study, I learned it very hard way. In fact, it is still I am trying to recruit my first patient in that study. that you must understand what you are promising. Patient, just because it looks good, patient may not be willing to participate in that study.
I had a last question was any other trial as a first. In short, what would have been your ideal first trial if you had the chance, like among your studies?
I think I am hindsight. I think I should have selected a ideal first trial as a phase 4 study. That should have been the way I thought as a first pharma study should have been a phase 4 study. But I have a few senior colleagues with me, and they said that you have a pretty good. But when I selected my first pharma study, I had already been a PI for 2 phase 3 investigative studies and a couple of phase 2 and phase 1. So they don't know you have experience enough and go ahead with it. But hindsight, I think maybe a phase 4 study would have been the best place to step in the as a PI for the pharma study.
So there are a few of questions which I asked for the audience. I mean, like our PIs, they have a lot of expectations. So there are few learnings like don't force trials on patients, performance status, try to avoid phase one studies. How much you agree? Harshil, you're there?
Yeah, Vinayak, thanks. In
So,
fact, I would like to just put the disclaimer that I have worked through quintiles, ICNOS Life Sciences, more from the sponsor CRO experience I've got. And when you ask me this question, it is funny because for a CML phase one first in human study, we had multiple, of course, it was a global study. And we had multiple centers from India participating. I think the reason why the PIs may be, what do you say, a little bit susceptible in participating is that the rigors of the phase one, the DLT, as well as the waiting of the patients for the cohort management becomes when we have a 3+3 design, they might have convinced the patient and suddenly the sponsor will say that I don't have the cohort ready right now, you'll have to wait or some things. And when they are entering at that particular period, we used to have either a blast crisis or some other complication, and then the patients would not be recruited. So the expectation mismatch between the sponsor and the PI, I think that creates a little bit of difficulty in terms of recruitment, as well as phase one as a most challenging thing. The PIs may probably give more information, but from the sponsor perspective, this is what challenge happens, how to manage the cohort. Thanks.
I was when I was a study coordinator in this year, there were ethical committees were not keen to have phase one, phase two study in a government setup. I mean, there was a lot of feedback. Vijay, is there same thing or it is better? Phase one, phase two study were not preferred.
Yeah. Can I take that?
Vijay,
Yeah, and I think sometimes I think what I have seen in my experience, even as an investigator for investigative studies, is that what you're saying is correct. Somehow our ICs are not that accepting for phase one studies, especially. It's like for multiple ethical hindrances come into place, saying that okay, there should be some more data. I think it is relevant from the PI side also, that there should be a better education for the IEC members also. That's what I feel, because if there is good data, then no one is going to go do a phase one study. Phase one studies are going to be done when you have limited amount of data, and safety is something which you are actually looking at. And that happens. And even what happens is that most of the phase ones have a good pharmacokinetic profiling going along with it, which requires multiple sampling to be done. There are issues from ethics, which I also have faced for doing such kind of pharmacokinetic profilings. But I think we should be doing more phase ones, because actually I like doing a phase one and phase twos more than doing a phase three. I think more science develops in phase one and phase two, even if they come negative. And it is difficult to do phase one. And I completely agree with what Dr. Harsha said. I have the experience because my team takes phase ones. So I was not a PI in that, but I was a COA in that. And it is very difficult that you counsel the patient, screen, did every study, everything is there, and you send a mail to the sponsor saying that this patient is ready and we are done, and let me know in which cohort we put it and what to do. And then it comes that the cohort has stopped and we are looking at the DLTs and let the patient wait for some time. That becomes a bit more frustrating. But I think, but that is the way at present phase ones run. So I think that is going to be a hindrance. But I actually enjoy doing phase ones.
Vinayak, Dr. Sahoo here.
Sir, a word, sir. We have Govind Babu sir also wants to add comment.
Yeah. So my point is, I have done 2 or 3 phase one in Kidwai as a study coordinator and 2 as a PI. I think without doing a phase one, you are incomplete as an investigator. And I totally agree to most of the point. It's one of the most challenging trials you'll ever do, but one of the most interesting. The problem what I faced is the ethics committee, because of the multiple pricks, what happens, and the amount of deadlock, which is insignificant. But the way it is sounded, and there are people who are layman and other persons in the ethics committee which you have to convince. So for me, I had to make amendment for a line or a port to be inserted for the multiple bricks also. So that was in 2009-10. So this is what I faced. But yes, I agree to Vijay that without a phase one, I think you are incomplete as an investigator. I'll use that word. It's lovely to do phase one, and it's challenging. It's not money rewarding. Even as a private investigator, you don't get that amount of money rewards as you get in a phase three. It's much more rewarding from the financial point, but this is more challenging. And I think all of us should do phase one.
Dr. Govind sir, want to add something?
Yeah, I totally agree. Phase one is actually what makes you a complete trialist. It's really exciting to do phase one. And what Vijay told is true. The ethics committee always has issues. So I think now it is mandatory that all these ethics committees undergo regular GCP training. So I think hopefully that will make some impact on them. Yeah, it's very difficult to convince ethics committees. And they say, how much blood you're drawing from my patient? You know, so it is difficult, but I think that is an exciting way of doing clinical trials. Absolutely.
Dr. Aditi, can you mention something?
Yeah, thank you, Vinayak sir. Basically, I do not agree with this that we need to avoid phase one studies. Phase one studies, I have experienced over the last 18 years. There are lots of investigators in our country, as well as in the audience right now, a lot of them who have done excellent job at phase one studies. And, you know, they are able to put India's name on the map when it comes to early clinical development for an innovative drug or for a biosimilar or generic drug. And what happens with these innovative molecules is when the publications are out and when there are presentations at ASCO and other similar forums for other therapy areas, our investigators have gone and presented the data. So there's no question of avoiding. Yes, one should, it's not about avoiding, but one should be very judicious in understanding whether the investigator and the site infrastructure and the site team are completely geared up to perform in a phase one study. Because at the end of the day, the responsibilities with the investigator is very, very great, that this can make or break a molecule and the conduct of the trial and the conduct of the phase one study. But and when it comes to ethics committees, I'm hearing this, but I've also seen ethics committees on the other side very, very maturely handling the phase one data. There are certain guidelines. ICMR guidelines has certain guidances for the ethics committee as well as the investigator, that what should be the blood loss which may be allowed. So I think, Vinayak sir, I would say the phase one studies, India has done a great job. And overall, over the last 18 years, yes, it's a very small population of investigators. And I agree that it should, everyone should try it, because being a part of a phase one study and presenting the data really enhances the skill set of an investigator. Thank you, Vinayak sir.
Thank you. So I want
Dr. Vinayak, sorry, I'll just make two quick points. One is that the DCGI mandates that 50% of the sites need to be government sites. So we will require cooperation from government sites, point number one. Second is that the phase one for NCEs and NBEs is only allowed for Indian companies. The DCGI does not allow the phase one of drugs which are discovered outside to be done in India. So these are 2 points which I just wanted to highlight for the sake of the audience. Thank you very much.
Yeah, that's right. That's right.
Yeah.
So, so in fact, a few of the Singapore and Canada have fellowship in phase one studies. In fact, I think one of the person who wanted to go to Singapore, and he's been paid more than a regular fellowship. Fellowship goes for BMT. So that is the virtue of phase one studies. So current role, I'll start with Vineet. Where you're placed now, how resourceful is your site management? Can you hear me, Vineet?
Yes, sir, you're audible.
How resourceful is your site as SMO?
Yes, sir. So I'm working at Artemis Hospital, Gurgaon, sir, and it's a corporate hospital. And thankfully, our research team is quite strong, actually. I think they have a lot of experience with studies, and usually they are a team of problem solvers. I think That is why things have been going easy from clinical trial point of view. So I'm quite happy with my research teams.
So how many active trials you have under now?
So at present we have, if we include all the PIs, we have got 3 consultants and all of them have trials under them. In oncology, in medical oncology department, we have 11 trials right now.
So this is just a question which ethics committee asked of the trial thing. How much quality time you able to? Hello, someone coming?
Hello. Yes, sir.
How much quality time you have for clinical trial?
Yeah. So as I said, so the responsibility is split between 3 consultants, and all of them are not PI for all the trials. So, for example, at present I am PI for 4 studies. So I think at present I'm able to give good amount of quality time, but of course it depends on person to person how many trials they can manage and in one go. So.
And what drives you for clinical trials thing? Is it you get a better use of the drugs earlier, or is it the patients gets sometimes not able to treat all the patients in corporate hospital? Sometimes you get a benefit to give a good treatment to even patients who cannot afford.
Yeah, so from my point of view, sir, I feel a clinical trial is win-win for everybody. Patient is able to get the best possible care. Often he's able to get almost all of his care free of cost. And in case of any toxicity also, patient can get the best management free of cost. At the same time, we are able to access the drugs which are new and, sooner or later, many of these drugs will become the new standard of care. So I think early exposure to the drugs is the big perk, and it actually feels good to be able to offer patient the latest standard of care. So I don't think there is any major limitation for patients to join a study, and that is what drives me.
Oh, Vijay, I think there are a lot of things which will not be there in a corporate hospital, which for a PI. So, what are your trial? I think I remember when you were telling electrotherapy, you were not keen to take it. I was like, why you didn't take it? So, in same context, what drives you your zeal for having clinical trials, pharma driven?
So what I think is that for me that it's one of my dreams, and I hope it is there, happens before I die, is that for every situation I want to see that there's a clinical trial wherever I'm working. So for every situation there's a trial for the patient, because what I think is that if some disease X has a 2-year survival of 30% with the standard treatment, however better standard treatment you give, you're going to achieve the 2 year survival of only 30%. If you want to achieve it 40%, 50%, 60%, or to increase this, you have to try something new. And the only way ethically and scientifically correctly you could try something new is by doing a clinical trial. So for me, that is the main source, because this is the best form of treatment that patient can get. This is the best thing which the best chance you're giving to the patient. Most of the trials will have a standard treatment arm, and most of them the standard treatment arm and something else. So this is the best chance you're giving to the treatment. So I think for the patient, this is the best chance. And as Dr. Vineet also highlighted, in our country, financials, we may say that financial issues should not be taken, but the practical part is financials are limited for most of the standard therapies for the patient. Like currently we have a phase 4 of cetuximab running. We may say that what great it is, but the thing is that still only 10% of my patient can take cetuximab. Because of that phase 4, at least all the eligible patients are getting cetuximab. So what happens is that allows the patients to take standard therapy and get a better chance of doing better results. Second is, as a investigator in the study, it gives me, I think, a head start to learn the way the new drugs have to be given, how do I monitor them, what are the common side effects, and I get a head start over my other colleagues, that I know more about this drug than other my colleagues, because I gave that drug when the study was going on. So in future, if it becomes standard of care, I'm well aware about how to handle those drugs better than others most of the time, because you gain that experience in that. So I like that.
Another question for you. Like, you are a big center with lot of consultants, with lot of trials. How your site is able to manage so many trials apart from the numbers?
So, so each individual consultant doesn't have multiple trials. We have a few trials, and we are restricted on pharma trials. So what we do is that we work as a group. So me, Kumar sir, Vanita madam, Amrit Joshi sir, and Nandini, we all work as a group. So we, even if it is like the trial is of, say, Dr. Vanita, I am very well aware about what is to be done in the trial, what are my responsibilities. If I am the PI, she is completely aware of what is to be done in the trial, and so that actually doesn't stresses out anybody. So if you are ill, if you're not there, you take care. And most of the trials, all trials are not actually recruiting very fast. There is 1 or 2 trials which are recruiting fast, 1 or 2 you are actually struggling to get the patients. So when you take when you have something like a group responsibility has been taken, it becomes easy somehow to even manage the patient well, even to document the things well. And sometimes you fall in some tricky situations about what to do next. I know that initial one of the immunotherapy trials which the patient actually developed jaundice during that, and we were not getting any reason why the patient got jaundiced. And I always remember this, that when we had this discussion, these were initial days when immunotherapy, and then actually we thought that it would be a lymph node blocking and the CVD. It was actually a inflammation of the papilla which led to the jaundice, and when we biopsied that papilla, it came to be only inflammatory cells. So then we actually thought back that it may be an autoimmune inflammation of the papilla which got us to jaundice. So when you are alone, you actually get a bit scared, okay, what this has happened. But when you are in, when you have groups, you have seniors, you have juniors with you, your colleagues with you, I think not only in trials, I think in every setting, if you work as a group, it makes things easy. It makes, individually you may think about 2 or 3 studies, but if you are like a group, then multiple studies you get to, and you get to learn and expose to other kinds of molecules also.
So I have another question, just to the persons who are responsible, the site coordinators, are same for your investigator-initiated trials as well as the pharma trials?
No. See, one of the things which we need to understand is that educate time needs to be there with the coordinator for doing the job which has been assigned to them. And if some trial is not recruiting, then I give some job to that coordinator just to keep them busy, so that they are a habit of doing some work. But if the trial is going properly, I don't give any job to them. So that's the way I deal with things. But no, I don't try to give one coordinator 2 or 3 jobs or 4 jobs, because then they make a mess of things. And then as Dr. Sahoo told us, then those mistakes start happening in documentations, those mistakes start happening in calling the patients, reminding them they need to come for visits, and then it's a mess which I actually don't like to handle after that. So it's better to prevent those things from happening.
Okay, I will. I'll ask Vineet. I think
Dr. Hemant wanted to ask a question.
Go ahead, sir.
Yeah, thanks. I have a question to Vijay. Vijay, is the number of trials per investigator fixed in Tata Hospital? There was, some dictate on this from the DCGI office. So can I know the current state? How many trials can a PI take? And is the number capped in Tata by the ethics committee?
Yes, sir. That number still remains the same.
What is the number? How many trials can a PI?
Five DCGI trials, not more than 5. So, but most of the time we don't try to exceed more than 3.
And you have a clinical research assistant, one clinical research assistant for each trial, or?
Yes. So we budget it that way, that every trial has we don't accept the trial if they're not going to budget for a person for that trial. It becomes very difficult. So before the submission itself, there is so much of paperwork to do. If you don't get that helping hand, it becomes difficult, sir.
And this includes investigator-initiated studies also, like 5 per PI?
So As said, there are no restrictions. So because the problem is quite a time the investigator initiated studies, multiple times there may be some retrospective or small prospective, and now with the number of students going up, most of the people have like two students. So at a time they may have 6 students. So they may have at least 6 student related research, because the student cannot be the PI. So that has not been kept as a limit, saying that because I know that few of our senior colleagues have two to three students per batch. So I don't think that is kept to the limit, because that many number of studies will be there at least for those students.
But the industry-sponsored trials is capped at 5 per.
Yeah.
I think that makes sense.
Yeah.
Can I make a comment also, Purvish? Do we have time, Vinayak?
Go ahead, sir. Go ahead.
So I think in the smooth conduct of trials, I have found that the cell phone is an extremely important instrument. And the PI, when you are starting the trial and talking to the patient and the attendant, should have at least 3 numbers for cell phone numbers of the patient and the near contacts and the household contacts, and the assistant should call these 3 numbers and verify that they are in existence and working, and these people are close to the patient and they can answer some patient related questions. If the patient only gives you 1 number, that always lands you into trouble. So you have 3 numbers listed in the case record file, and all 3 numbers have to be called on and verified. That is like one tip for the new investigators, that you make sure that your cell phone communication, there are 1 or 2 backups to that. And the second thing which I'd like to tell the new investigators is that if you're doing a chemo trial, then I think once you've administered the chemo, particularly to the trial patient, have your assistant call that patient every alternate day till the next follow-up is due. So that gives some security to the patient, that opens up communication channels, and that picks up some serious adverse effects very quickly, and you can intervene at that time. So just make sure that your assistant calls the patient or his assistant and documents in the case record file. That helps big time in opening communication channels with the patient and also picking up SAEs early. Thank you.
I'll just extend Hemant's comment. That is a very good way of doing it. In fact, in each follow-up, you should actually keep asking that whether these 3 numbers are still working or not. Because nowadays, with the mobile being okay for all India numbers, mil jata hai, so they keep it. But in old days, when the number used to be like Bombay specific or Maharashtra specific, patient like in Tata used to come, take a SIM card from Mumbai. That time it used to work, but after he goes back to his native place, it is to change. So it's always a good idea, even in follow-up, just to confirm that these 3 numbers are still working.
That's a good suggestion. Actually, apart from clinical trial, it will work for practice also, because one call of post treatment, they would actually be really happy, because I would like to get that call. So I have another few questions. We'll stop at around 9 o'clock, whatever questions are left out. So Vijay, for you, how did a CRO contact you for feasibility, or anything makes you to choose a specific CRO or avoid any CRO?
No, I don't. If I find the trial interesting, I'll take it.
We'll ask.
Of course, if I find a trial interesting, I take it. Of course, I discuss it with my colleagues just to see that we have a few investigator-initiated trials in. So I need to keep that balance, that there should not be multiple competing trials for the same patient. So we discuss among a group, anyone who wants to take the trial, if it's fine, and if everyone is okay with that, then we go ahead with that.
I would ask someone who's not diplomatic. Vineet, do you feel that you will avoid any specific CR or something like that?
No. So maybe luckily, or because my experience is not so high, I have not had any such bad experience that I have to avoid any CRO as of now, sir.
Govind Babu sir or Purvish sir, can I take this thing?
So I think it is. Can you hear me?
I can hear you, sir.
Yeah. So rather than a CRO or a sponsor, it is the people that matter. If there is a possibility to have a dialogue, and if it is a possibility to understand each other, then it is worth pursuing. Sometimes if the protocol is good, I'm interested in it, but the representative of the sponsor of the CRO who is not understanding or as dogmatic about certain things, then I would not do that.
There are issues in CRO where they keep on calling and trying to put the pressure back on you. We don't have any obligations. We have told some number. For most of patients' number, I can start with 0 to 2. I always keep little 0 actually. 0 to 4 or 5. I never give a number because that makes me say that I already told these indications were not there. We have numbers. So I think Vijay's formula is actually better than my formula, where I used to do a half of everything. But that's the reality actually. So this is the same thing which has been told.
So Vinayak, I think the situation is absolutely different at a huge center like Tata and Vijay, no? You don't have any problems ever with numbers. At smaller centers and more modest centers, you have this problem. So TMH SOPs with regards to numbers don't apply to most centers.
I agree with you, sir. There is post your big institute leaving, no, will always lose that addicting power of deciding so many patients. What day will be making difference to so many patients, which will always miss it. So
when they're talking about competitive recruitment in a trial, and then I always want to know if TMH is part of that trial or not. If TMH is part of the trial, then 90% of patients they will recruit, and before you have your first patient, the recruitment is closed.
That's a good way to look at it.
Sir, don't worry. At IC, make sure that we are not the first center which opens up. We take at least a year after the first center opens up, so that everyone gets chance to recruit patients.
But I found it interesting that there are some centers in India who get a very good numbers within short time. I'll not take names, but there are centers who are well tuned. In fact, I went to an investigator meeting. They were telling the trial eligibility is assessed from the registration. So if there is a lung cancer trial is running, there will be a insight that can it be taken for trial. So that kind of thing also I've heard about it. And that institute name is always on the top recruiter. I have seen that also. So, but coming to as a science, what are the scientific values you look closely to select a trial or not to select a trial? So you said interesting trial. So what makes you interesting? Phase. You can take on all the listed topics which makes you better. So start with Vijay, then I'll go for Harsha and then Vineet.
So, phase of the study. So like if already I have 1 or 2 phase 1s running, then I don't jump for the third phase 1. One phase 1 running makes you bit it is quite resource consuming with respect to the time. I don't like when the study protocols are too complex. I like the protocols when they are more realistic. Like this, as I told you, the first protocol which I'm yet to recruit, which is going on since last 3 years. Well, for 1 year, the patient has to stay in Bombay and take treatment. Such then patient recruiting approach. Numbers are not an issue at our center. It's more of about logistics for the patients. Like, can they travel and come? Can they stay near and come? That becomes, because now, at least in Tata, what is happening is that 50 to 55% of patients are out of Maharashtra. And per se from Bombay, we get only around 15 to 20% of patients. 30% is Maharashtra. That has been a breakup. So, so to tell the patient that they need to be around, that feasibility we need to get assessed. And yeah, reimbursement, it has to be very clear that what they are going to give, and it needs to we discuss that a lot. And competing studies, yes, we need to think of what I meant by competing is that if we have our own study running in that situation, then the preference goes for an investigative initiated trial and not for a pharma study. So we would not, if there is a investigative initiated trial going, it would get the preference over a pharma study. That's the way we look at it. And luckily the people around me, if the science is good, if the question is good, the protocol looks good to us, And if there's no competing results, we normally take the study. That's quite motivated people who surround me, and hence motivation is not an issue with at my place.
Dr. Vineet, do you disagree with few things or you agree with most of things Vijay said?
I agree with most of this. And this all things need to be looked at before you take a study.
We'll ask from CRO's perspective, Dr. Aditi, ma'am. Do you target PIs or sites based upon the design of the scientific value, or how is it, Dr. Aditi?
Yeah, so Vinayak sir, thank you for the question. One of the most important things, like if I have to look at, there are various other points whereby we do investigator selection, but if I have to stick to your slide, then what happens is that certain phase of the study, like for example, if it's phase 1 or phase 2A, we would stick to certain centers and focus more on centers of excellence. And like Purvish sir said, that it's also about how we have a relationship with the investigators where that comfort level is there, and where you can have a very scientific discussion with the investigator and take actually guidance of investigators when the study protocol is being developed for complex studies or for the early phase studies, because this is very important. And the other thing that we would also look into is we'd never look at investigator grants. That is the first thing. We don't look at that. The other most important thing which we look at is the kind of infrastructure and the support staff that the site team has, and the motivation of the investigator to do that particular trial. Because if the investigator is scientifically or otherwise convinced and motivated to do a trial, then the performance is very different vis-a-vis, the investigator saying yes to a trial based on a relationship or based on some other pressures. So these are I would always give, these are my personal views, and this is how I've done research since 18 years. I would give importance to the investigators' feedback on the protocol, number one. And how excited they are about it, because the minute they believe in the scientific, concept, the rest falls into place more or less, and which we can make it work, and we need to make sure that we partner with the investigator to support them to come up to speed, and not just have a checklist that, okay, if they meet yes or no, and that's it. What are things whereby we can develop a site or an investigator? That is something also that we look into, sir.
Thank you, ma'am. Harsha, anything to add or?
Yeah, thanks, Vinayak. I know the time is less. A couple of points. One is the phase of the study, and depending upon the site, the match has to be there. Then there is a feasibility questionnaire, which is normally sent to the sites before these studies are kind of started. And third, today, most CROs have risk-based management as one of the monitoring aspects, and that kind of gathers a lot of data regarding each site, from the recruitment speed to the data cleanliness and the quality aspects. So if a particular site is not adhering to certain, FDA expectations or the regulatory expectations, then those sites are not reached out to in the future. So data is coming into play at multiple levels. Most big CROs have data on each sites, and then that data is taken into factor along with the feasibility studies. That's from my side. Thank you.
So other question in the same context, few of the investigator, few of the sites ethics committee have more very stringent on protocol deviation. A lot of things they take very, it's good to take independent view. Sometimes it may be not be right view. I think we had a discussion with Sahoo sir also. So those do matter to you to reach out for the PIs or the sites. Harsha, I can take on.
Sorry, yeah, ethics committee clearance is factored in from the starting perspective. And since we know that few sites will not start early, if the ethics committee is going to be stringent in nature, but later on, if the ethics committee is active, it actually helps us from a perspective that ensuring the safety of the patient, and therefore we do not, in sense, take that as a peculiar challenge in terms of looking at a site. I hope that answers your question. Yeah.
Most of the trials will happen in 50% in government setups. So that is the norm there. All ethics committees are not same. I mean, there is an NIH accreditation for ethics committee now as we speak. It has to be accredited. So some kind of ongoing process. It has to be accredited every 6 months, 1 year online. We had a virtual ethics committee visit, some accreditation. So with that, I think uniformity is achieved, or we have some variability.
I think I believe there's still some time to go with respect to uniformity. It is not still as uniform as one would want it to be.
So can I just make a statement here, Vinayak? It is impossible to have uniformity of ethics committee because the local circumstances may be totally different. For a center which is a government hospital, the infrastructure may be different from what is there in a state-of-the-art hospital. So I don't think we should ever expect that there will be uniformity in the ethics committee action. What we need is uniformity in their level of competence and training about what is their role in the ethics committee. That's what I wanted.
Agreed, sir. Vinayak, sir, can I just make one comment? Just one. I know we're running short of time, just 30 seconds. I'm actually an NABH Ethics Committee Accreditation Assessor. I've taken the training for that. And related to your point, I completely agree with Purvish, sir, because if you see the NABH accreditation process, the entire, if you go through the criteria, and it's more focused on a process and procedural. It is not really meant to automate one EC to the other. And every ethics committee constitution and people come up with different views, and rather we should be more inclusive about different views, and ethics committee should be more patient centric. So the NABH part will only be about procedures and documentation and processes and trainings and things like that, and how an ethics committee should ideally function. It doesn't have any intellectual intervention from a standardization perspective, Vinayak sir.
Thank you. In fact, I was trying to, the doctor's part is still reasonable. The non-medical part, they need to be constantly be updated, because the NABH accreditation really helped our center when we did a repeated revisions of SOPs and other things. It got little kind of, we were clear with most of the guidelines. So I'll take the last things. I'll start with Vineet. How much patient recruitment weigh in trial selections? Like, I think Vijay already has spoken. Vineet, can you hear?
Yes, sir. So, sir, for me, it is important. Like we discussed earlier that at the end of the day, you also have limited amount of time, and your research team also has limited amount of time. So you can't just, take every study. So sometimes there are studies where even if the hypothesis is important, I know that I will not be able to give more than maybe one patient in the entire year. And even that is a question mark. So I will not take a study of that nature. For me, I think that I should have the possibility of giving at least 2-3 patients per year. Only then generally I will take the study, because of the, at the end of the day, whether you give a patient or not, it involves a responsibility and a time commitment also.
Thank you. Vijay, I have this question for you. I think you were trying to recruit patients for one of the trial, and I got a mail also. So what are the things you were able to do to increase the visibility of trial at both at local Bombay or maybe outside your site? I will mute Vijay. Vijay, you're mute.
So that mail must be for the meningioma trial, which is not recruiting that well. What I think is that as PIs, and I think people who are involved with research, I think it is important for us to even advertise that which trials are going on. For example, like there might be a patient with me, and if I give that, there might be a good trial which might be going out in a corporate hospital. And I don't mind sending my patient for that trial if the patient is eligible for that. But quite a few times when you tell patients that there is a study and they at least want to know that, okay, roughly that whether the patient is eligible or not. So I had this discussion, especially in gliomas, when few of the patients who have relapse and actually want to go to US or Singapore, because there are not many relapse glioma trials which are going on with us. And that time it becomes easy to talk to the PI, see that whether it's roughly eligible, the patient is eligible or not. So I think that's we need to advertise trials, and especially in local, like in Bombay, I will make sure that trials which I'm doing and whom I anticipate that I won't be recruiting as my intentions are, I would let my colleagues in Bombay know, colleagues around Maharashtra know for such kind of studies, which I anticipate if there is low recruitment. But I think the good policy would be that all studies which have been done by the PI Investigate Institute of Pharma, we should have a platform. The CTRI gives a platform, but I think there should be another platform where you know that, okay, lung, this situation, these are the studies running and these are at this center, so that a quick look even for the oncologist and even for the patient can help if you want to give that option of a study for the patient.
So I have another question which relates to you also. When patient is on trial, goes to a native place and has some SA or A, so how do you
So this is a practical thing which happens, and this happened a lot during the COVID time, that the patient goes to and has a SAE. Then in that case, I think what Dr. Sahoo said is correct, that you are going to face this situation. I think the first thing is that within 24 hours of you coming to know that the patient had an SAE, it may be happened 15 days back, but you came to know, like if I come to know now at 9 o'clock, if I get a call saying that this patient had an SAE, within 24 hours, you should intimate your IEC. And if it's a DCJ register, then it needs to be go to the head of the institute and to the DCJ also. And then you start the process from that. Okay, whatever minimal information you have been able to gather, you at least make a summary and intimate to the and then you call up the patient, and if the patient had got admitted somewhere, what happened, if you can get the records and make a summary, and then that's the way I do that. It happened a lot during the COVID time actually.
I think we will 9 o'clock is.
Yeah, Vinayak, I think it's past 9.
I think we close it off. I want to close the session. So
thank you very much, Vinayak. Yeah. For those who are interested in getting the certificate, we have put the link for the final MCQs. Please answer those MCQs. You will automatically get your score. And if you get a score of more than 60%, you will get the certificate. We thank all the faculty and thank Vinayak for being such a wonderful program director. Over to you, Vinayak, for the last word.
Thank you, sir. I would thank all the speakers and faculties for a wonderful session. I would also thank Kashish and other organizers who have done a great helping hand for me. I didn't even feel the pressure of it. And lastly, but sponsors of the sessions, Lance Torrent and SI, who have been supported us. And thank you, everyone. And good night. Maybe we'll see you.